Hi, I'm Kavir Ramdass, and you'll follow me on the slides as I brief you on this article by Chow et al. This article investigates the differences in color Doppler image mapping between free jets and surface adherence jets as a result of the Coanda effect. Now, the Coanda effect was first noted in 1910 by Henri Coanda in the field of aeronautics. As you can see here, when we have a jet where the outlet is very near to a surface, the jet has a tendency to adhere to that surface and follow the contour of that surface. This is as a result of an asymmetrical shear layer we have developing between the surface and the adherent jet. So this is the adherent surface, and this is the first layer of molecules in contact with the surface. We know from fluid mechanics that there's a no-slip condition, whereby this first layer of molecules has a zero flow velocity relative to the surface. This in effect causes a viscous drag between subsequent layers, which causes the fluid flow to be pulled towards the surface and along its contour. This is what contributes to the Coanda effect. In modern medicine, the Coanda effect is very relevant in Doppler radar and imaging, which is used to visualize blood flow throughout the heart. It's used as a quick diagnostic tool in detecting valve abnormalities such as mitral valve insufficiency and pulmonary valve stenosis. They do this by examining the cross-sectional area of the jet ejected through the blood vessels. And this is where the Coanda effect comes into play, as it causes a drastic reduction in jet cross-sectional area versus a free-flowing jet, which has a much larger cross-sectional area. It also causes retrograde flow or reverse flow, as well as variance flow or chaotic flow, which can result in a misdiagnosis. Another example we see here, as a result of supravalvular aortic stenosis, we see that there's a convex surface being created at the narrowing end here near the orifice. And this causes the jet to adhere to that surface via Coanda flow and run along the surface and result in an asymmetrical blood flow on the upper extremities of the body. They modeled this in vivo system in vitro by creating a closed-circuit fluid system using a continuous flow pump to drive fluid through an outlet into a receiving chamber. At the outlet, they established 4 different surface conditions using a smooth aluminum surface which was either straight or curved. The first 2, the surface was displaced from the jet by 4 millimeters. 3 and 4, they were at the point of incidence of the jet and a free jet as the control. So they used a Doppler scanner and an RGB analyzer for 10 different flow rates in each of these 5 cases to find the average cross-sectional area in each instance for each flow rate. They used an image histogram similar to what you see over here to extract raw RGB data for image pixels in the modeled flow, and with that they were able to determine the average amount of red, blue or green pixels in each case and the average intensity and location of these pixels. This allowed them to quantify desired flow in the red, chaotic or variance flow in the green, and retrograde or reverse flow seen in the blue. They used the histogram to compare the variance flow and the retrograde or reverse flow on the surface side of a Coanda jet to the corresponding side of the control jet for the same flow rate. They also compared these factors on the surface side of the Coanda jet to the corresponding side on the same jet. After analyzing all of this raw data, they found, as expected, that the jet with the largest cross-sectional area was in fact the control jet. This is because of something called mass entrainment. This happens when a free jet enters a chamber. Here, it creates a mutual viscous drag layer between the fluid already in the chamber and itself. So as a result, it dissipates its kinetic energy by kind of dragging along fluid from the chamber, and because of this it widens and spreads out symmetrically in all directions. The smallest cross-sectional areas occurred where the surfaces were placed at the outlet for the nozzle. This was particularly apparent in the curved surface, which showed the smallest cross-sectional area of all. Where the surfaces were displaced 4 millimeters from the nozzle, the cross-sectional area lay in the middle between the control and conditions 3 and 4. They also confirmed that variance and reverse flow on the surface side of Coanda jets was substantially higher than the corresponding side of a control jet to the same flow rate. The main conclusion was that the prime variable determining cross-sectional area was the distance of the orifice nozzle from the surface. Limitations to the study included a limited bandwidth from the recording apparatus and the fact that no high-velocity flows were measured. Drawing from what was learned in the study, it's prudent that areas having more laminar flow characteristics such as source chambers rather than receiving chambers be used in the future for Doppler flow analysis to ensure accuracy. Thanks for your attention, and even if I'm not selected, I hope you appreciate the time it took to make this. Thanks very much.